biology and artificial intelligence. In a contemporary society where the understanding of the genetic code has been demystified and the distinction between the digital domain and the fundamental nature of living organisms becomes increasingly indistinct. A compelling narrative emerges, urging us to assert our agency and assume command before we are overwhelmed by the relentless wave of technological advancements. In his seminal work titled The Coming Wave, Mustafa Soliman urges us to explore a frontier where the realms of synthetic biology, artificial intelligence intersect, enabling the manipulation, revision, and reimagining of the fundamental essence of life itself. However, as we find ourselves on the cusp of this forthcoming period, we are confronted with the pressing issue of containment, which compels us to exercise caution and prudence. Are we adequately prepared to negotiate this unfamiliar and unexplored territory? Please join us as we embark on an exploration of the concepts of discovery, possibility, and caution within a society where evolution is no longer a natural process, but rather manipulated by intentional design. The commencement of the expedition was marked by the initiation of the Human Genome Project. A monumental endeavor focused on deciphering the intricate genetic blueprint of the human species. This effort not only facilitated the comprehensive deciphering of the human genetic map, but also laid the foundation for a multitude of developments in the respective sector. In the examination of the economic terrain, Moore's law, a prominent concept forecasting the doubling of transistors on a microchip every 2 years, has garnered significant attention. However, the Carlson curve, which demonstrates the rapid decrease in expenses associated with DNA sequencing, is often overlooked. This reduction has been remarkable, with a decrease of 1 million times in less than 2 decades, surpassing the rate of improvement predicted by Moore's law by a factor of 1,000. With the decline of cost barriers, the field of genetic engineering has witnessed a significant expansion beyond the capacity to only decipher genetic code. Presently, it empowers us to not only interpret, but also modify and create genetic code. The pivotal moment occurred in 2012 when Doudna and Emmanuel Charpentier introduced the CRISPR Cas9 technology. In recent times, the process of gene editing has become far more accessible, resembling the manipulation of text or computer code in contrast to the initial stages of genetic engineering. The potential impacts of germline cell editing are significant, particularly in relation to its effects on evolutionary biology, as alterations made in these cells can have far-reaching consequences that persist across multiple generations. The user's text does not contain any information or context to be rewritten in an academic manner. The progression of the tale encompasses the emergence of DNA printers and the field of gene synthesis. Organizations such as DNAscript are leading the way in the field of genetic engineering, where they engage in the production of genetic sequences using a process akin to printing DNA strands. This pertains not only to the replication of established genetic sequences. The topic pertains to the creation of novel entities, namely, the manipulation and design of living organisms. The current paradigm shift in biology, emphasizes the central role of focused design in the development of self-replicating, self-healing, and adaptive processes that exemplify the principles of evolutionary design. The applications are unlimited, ranging from viruses modified to make batteries to algae that pull down carbon from the atmosphere. The field of systems biology seeks to comprehend the holistic perspective of cellular processes. The utilization of bioinformatics and computational biology enables the comprehensive examination of an organism's functionality at both the tissue and organismal levels. These endeavors have the potential to serve as the cornerstone for a novel age of individualized medicine. The provision of healthcare services and the selection of medications would be intricately customized based on individual DNA and unique biomarkers. Altos Labs is a company that is committed to revealing highly effective solutions for combating the aging process. The company is dedicated to the pursuit of reversing the aging process in humans with a specific focus on rejuvenation programming techniques. Their primary objective is to reset the epigenome, which consists of chemical modifications on DNA that regulate gene expression by activating or deactivating them. As individuals age, there is a tendency for these positions to become inverted. The objective of this experimental methodology is to revert the aging process, thereby enabling individuals to surpass the average lifespan of 100 years. This achievement is anticipated to be realized in the near future, specifically within the next decade. It is important to note that the primary goal is not solely to prolong human existence, but rather to reverse or halt the aging process. The focus of this study is on improving the overall well-being during the aging process. Sustainability is a crucial discourse, and the field of biotechnology presents opportunities for the development of environmentally benign methods for producing materials such as plastics, cement, and fertilizers. The method developed by Nobel winner Francis Arnold, which exhibits a 15-fold increase in energy efficiency, serves as a compelling demonstration of the promise of biotechnology in the creation of sustainable solutions. The emergence of biofuels and bioplastics serves as evidence of this transformative shift. As we contemplate the future, the implementation of automated scaling in genetic engineering procedures has opened up a domain in which a wide range of biological materials and structures can be created with little resource inputs. If enhancements can be achieved in both the articulation of DNA and its subsequent utilization for the purpose of genetically modifying a wide variety of novel organisms, the automation and scaling of these procedures could potentially enable a device or a collection of devices to generate an extensive assortment of biological substances and structures while relying on only a limited number of fundamental inputs. In the future, it is conceivable that computers could be developed by a process of growth and manufacturing. It is important to note that DNA represents the most efficient data storage mechanism now known to us, surpassing contemporary computer systems in terms of data storage capacity, density, fidelity, and stability. In theoretical terms, it is postulated that the entirety of global data may potentially be stored within a single kilogram of deoxyribonucleic acid, DNA. The discourse surrounding the issue of containment highlights the significant importance of having a well-structured response system, particularly in light of remarkable advancements in technology. This discourse emphasizes the importance of actively engaging in debate and taking action to ensure the vast potential of synthetic biology is guided by ethical considerations and a sense of responsibility. Thanks for watching. Please do not forget to subscribe this video.